All right. So the discussion today, we're going to split this up over two separate lessons or two separate lecture periods with the endocrine system. So pretty much the first half is going to be the physiology of the endocrine system, functions of the endocrine system, different ways that we can utilize chemicals as signaling molecules in our bodies. And then we'll end with a discussion on how we can use hormones to kind of control the production of other hormones, and then utilizing the two major pathways that hormones can act on target cells at the end of today's lecture. Let's see. Participants, that's the one that I want. Let's see. There we go. That's what I wanted to do. Yes, Pamela, I'm reading your message right now. Okay, sounds good. Just kind of work with what you can. All right, so just to kind of give you a sense of how intricate hormone control is the beginning of your textbook opens up with a case study of something called androgen insensitivity syndrome. So this is interesting because this will make a lot more sense once we get into reproductive physiology and we talk a little bit more about how sexual phenotypes are expressed. Sexual phenotypes are expressed through genomes, and the traditional kind of most common forms of sexual phenotypes are having either an XX chromosome combination, which would result in being physically or phenotypically female, and then the traditional XY chromosome combination where you would result in being phenotypically male. So traditionally, the Y chromosome carries a gene on it called the SARI gene. Now, the SARI gene carries with it a whole sequence of instructions for molecular pathways that cause the developing androgen glands, so the adrenal glands, to produce androgens to trigger the development of secondarily sexual male traits. So what's interesting is sometimes there's a condition that'll develop in the body called androgen insensitivity syndrome, where the body's organs become insensitive or non-responsive to androgen secretion. And what'll happen is the body that has an XY chromosome combination will develop as being physically or phenotypically female, because the natural human condition without the SARI gene in place is to look phenotypically female. So this individual that's at the beginning of the text is cited with, going through puberty, but not actually starting their menstrual cycle. So they end up going to the doctor's office, and the doctor runs some blood work on her and runs some tests, and then they, figure out that this individual has androgen insensitivity syndrome and actually is genetically male, but looks physically female. So it's an interesting combination of how, with the lack of proper hormone production, that can trigger a result of a physically different phenotype that's contradictory to the genotype that this individual carries. So it's very, very interesting, and all kinds of issues can pop up with hormones. This is just a very interesting kind of in-depth example of how, if the body isn't responding to androgens the way that it's supposed to, or if it doesn't produce them the way that they're supposed to, androgen insensitivity syndrome can pop up. But this is more specifically when the androgens are being produced, but the body just doesn't respond to them the way that they're supposed to. All right. So as we've kind of already alluded to, hormones are going to be responsible for a wide variety of what we call long-term responses, right? So things like growth, reproduction, development, and then things like homeostasis are controlled in part by the endocrine system. Okay. So we use chemical messengers in the form of these hormones to transmit information or signals between different tissue types and different cells. And so we can do that in a few different ways. So we've got a couple different classes of chemical messengers. We have autocrine chemical messengers that get secreted by cells within a local area and influence the activity of the same cell type or other cells or the same cell in which the secretions were produced. So things like eicosanoids, prostaglandins, thromboxanes, prostacyclins, and leukotrienes, all chemicals that get released by things like platelets as a response to a clotting event occurring. So if you get like a cut or some sort of laceration and a blood vessel gets ruptured, then we have all these different kinds of hormones and other chemicals that'll get released by platelets to trigger the adhesion or stickiness of other platelets in the area to create a platelet plug before the actual clot can end up forming. So autocrine chemical messengers are kind of these localized chemicals that get released by a particular cell type and will only affect other cells of the same type in that local area or the same cell that it was produced by. And then we have paracrine chemical messengers, which can be produced by a wide variety of tissues and get secreted into the surrounding extracellular fluid and can have a localized effect on not only that same cell and the same kind of tissue, but other tissue types within the area. So our classic inflammatory response would be a good example of this. So chemicals like histamine, for example, super, super common, most often produced in every single inflammatory response that our body produces. So autocrine and paracrine are interesting because autocrine, auto meaning self, para on the periphery, on the surroundings, like immediate local surroundings. So if you're looking for a little acronym to kind of help you remember those, that's what I usually use. So neurotransmitters are ones that we've already talked about and discussed and that you guys have already read about. Neurotransmitters get produced specifically by neurons, and they will be released via the presynaptic terminal or the telodendrite, and they can end up passing across the synaptic cleft and then will affect target structures in a variety of different ways. Sometimes those neurotransmitters also, which can be categorized as neuromodulators, if they have much longer lasting effects, will either affect a neuron that's the subsequent neuron in the sequence chain or downstream, or if that neuron is synapsing onto a target organ, then the target organ will have a response to those neurotransmitters, right? And then we've got our endocrine chemical messengers. So these ones are the ones that we're going to focus on mostly today. We'll actually talk a little bit about neurotransmitters too, but endocrine chemical messengers get secreted by specialized glands called endocrine glands, which, remember, these ones don't have any duct system. So there's no tubes or pores that these hormones get produced into or out of. They end up getting into the circulatory system, into, I should say specifically, the cardiovascular circulatory system by way of rich saturation of capillary beds surrounding each of these glands, so that when they produce the hormones, there's really nowhere that they can diffuse but into the cardiovascular system, right? So we've got all kinds of hormones. We're going to talk about most of them today, or I'm sorry, not today, but probably on Tuesday. Things like thyroid hormones, parathyroid hormones, calcitonin, melatonin, growth hormone, luteinizing hormone, prolactin, progesterone. The list goes on and on. We could sit here and name them all, but that wouldn't really do us much good yet. So in essence, all of these hormones are going to enter the blood supply and be able to move to whatever kind of target tissue that they need to get to, whether it's in the immediate area, whether it's the bloodstream itself, or whether it's a target tissue like the intestines or muscle or the heart or whatever it is. They can move throughout the blood supply to be able to affect or latch on to those other target cells. All right, so here we've got most of our endocrine glands being represented. So these are not the only structures in the body that will produce hormones specifically, but these are just some representative glands that produce lots of hormones that are going to be our focus for our class. So as I've already mentioned before, once again, functions of the endocrine system, metabolism, regulating how we process nutrients and food. This is something that we'll get to once we get to like nutrition and metabolism and talk a little bit about things like personalized nutrition, which might actually in this day and age be more effective than just kind of, abiding by like the standard FDA, rules for, food consumption, grain consumption, whatever it is. Nutrition is important for everybody, and hormones are an important part in regulating how we, process those nutrients in the form of our metabolism, right? So making sure that we can, have, appropriately functioning metabolism. Come here, right? You need to just relax and just sit and not fidget. Sorry, my cat here is being super fidgety, and she just wants to kind of turn around and snuggle, but she just needs to, you just gotta relax, okay? You just gotta relax. Okay, so control of food food intake and digestion. So whether or not we feel hungry and we want to eat more is all controlled by hormones. You might notice too that if you eat slowly, you end up getting full quicker than if you eat faster. It takes a little bit longer for you to get full, or I shouldn't say longer, but it takes a little bit for your stomach to catch up to, or your brain to really catch up to what's going on in your stomach. So by consuming food at a slower rate, usually I think the rule is like 20 to 30 chews per bite of food. What is like kind of the standard for food consumption, but you allow for your stomach to expand. And when your stomach expands again, so the stomach isn't listed on here, but your stomach will produce hormones that your brain recognizes like, oh, there's some stuff in here and we need to take some time to break this down. So then we'll send other hormone pathways through the intestines and satiety centers of the brain to let your brain know like, oh, we're actually full and we don't want any more food. So again, food intake, whether we feel hungry or not, whether we're thirsty or not, all of that's controlled by hormones. Tissue development. One of the main hormones that's responsible for tissue development is growth hormone. So growth hormone is kind of cool because it pretty much acts on everything in our body. It's produced by the pituitary gland, and it's responsible for developing most every single tissue in our body. But then, as we talked about at the beginning of class, we have other hormones like androgens that are produced by the adrenal glands that sit right on top of the kidneys. Those will cause other tissues to develop as a result of things like puberty and age, and as we progress through life, right? So tissue development, what else? Ion regulation, water balance, all of those are kind of wrapped up in hormones that get produced within the pituitary gland. Things like vasopressin or antidiuretic hormone will control things like how thirsty we are. We have hormones that get released from the kidneys themselves that'll control how we respond to salt content in our body, and how we can regulate different kinds of ions like sodium, potassium, calcium, chlorine, all of that through our kidneys. All of that's hormones that get released by things like the kidneys. So the kidneys are depicted on here, but they're not listed as like an actual endocrine gland. The kidneys do produce hormones. And I guess too, that's another thing to recognize here, is most of the glands that we'll discuss, there's other hormones that get produced by other glands that aren't discussed in the book. You won't be tested on them because they're not in the textbook. So, if you're looking for something to go by what the book says with that too. Heart rate and blood pressure, controlling how fast our heart needs to pump, if we need to control our blood pressure by producing more urine or, increasing fluid intake. Again, all of that is controlled by hormones. Blood glucose and other nutrients. You guys get the picture here. Every single one of these functions that's listed on the left-hand side is going to be controlled by the endocrine system and hormone production. It's kind of crazy how simple chemicals that can be released into the bloodstream and then act upon target tissues, very similar to like a lock and key mechanism, how many effects they have on how we kind of run our daily lives. There's some interesting, just books for the public that are out there that look into really how pervasive the control of hormones is on our body. You guys should check them out. I'll try and find the book. There's an endocrinologist from Duke University, who published a book on basically how hormones kind of like run our lives and influence things like decision making and how we perceive different kinds of morals and ethics and things like that. It's very, very interesting. I'll see if I can grab the title for you guys and I'll post it on Canvas later. That's pretty cool. But the name of it escapes me. Let's see. So yeah, you guys get the idea here. Reproductive function control, uterine contractions, milk letdown, milk production, mammary gland development, immune system regulation, everything. The list is huge. But these are just some of the main functions of the endocrine system. Okay, so the nature of hormones is that they're chemicals, right? And chemicals get a bad rep because of, media and putting spins on things like chemicals in our food and chemicals in this, chemicals in that. Not all chemicals are bad, right? Our bodies are made of chemicals, okay? There are certain chemicals that are bad, sure, absolutely, but not all chemicals are bad. For example, our chemicals that control pretty much how we live our lives, our hormones, come in two different forms. They either come in fat-soluble or lipid-soluble forms, or they are water-soluble hormones. So typically the fat-soluble ones, things like steroids, things like testosterone, estrogen, those kinds of things, are what we call lipid-soluble and require transport proteins in order to make their way through the bloodstream. Because believe it or not, our blood is actually a polar mixture. And so things that are fat soluble don't really tend to dissolve well in the bloodstream. So fat soluble hormones like cholesterol, estrogen, testosterone, those kinds of things require transport proteins in order to get to wherever they need to go. But once they do, the way that they affect their target cells is pretty interesting. It's going to be different than our water-soluble hormones that can dissolve quickly in the bloodstream. Some of them require transport proteins, but others don't, because again, the bloodstream is polar. It is a water-soluble environment. So anything that can dissolve in water will dissolve in our blood. So these guys tend to be a little bit smaller. Good examples of this are things like insulin and glucagon, which can dissolve within the bloodstream and then regulate things like blood sugar content, right? So we can use these two mechanisms of dissolution is not the right word, but these two mechanisms by which we can kind of store and transport hormones throughout the bloodstream to have them get to target structures and wherever they need to go most effectively. So we've got a couple different ways that the release of hormones gets activated or controlled. So we have humoral control, which is basically regulation of chemicals in the bloodstream. So if we have certain chemicals that are normally floating around in the blood and then those drop or elevate, then we can release hormones in response to that effect. Then we can also have neural input where we have direct nervous system stimulation of an endocrine gland to release a particular hormone into the bloodstream. And then we've also got hormone on hormone regulation where we can have the production of hormones in one gland that will then activate the production of hormones in another, and then sometimes, in some cases, even go to another gland to activate other hormones. It's kind of trippy. But so those are our three main ways that we can control the production of hormones. Humoral, which is fluid concentration. So how much stuff do we have in the blood supply that's going to determine whether or not we need to release hormones. Sometimes it's a direct reflection of the concentration of that particular hormone. So like, let's say, for example, if we have insulin in our bloodstream, if we have enough insulin in the bloodstream to where our blood sugar drops below a certain threshold, then the body will recognize and say, oh, okay, we've got enough insulin in the bloodstream. We don't need to be producing this anymore. So then we can break that down, metabolize it, recycle it, and then reintroduce that back into the pancreas. So then we don't have as much insulin in the bloodstream, okay? So humoral, fluid-based control, neural, nervous system-based control, that one's fairly self-explanatory. And then the hormonal control, where we can have dedicated hormones that will activate the production of some, or some that will inhibit the production of others. And what's kind of funny is some hormones will actually inhibit themselves by production. So it's kind of cool. All right. So we've got, as I mentioned, lipid and water-soluble hormones. Why some of this matters for medical applications, right? Lipid-soluble hormones can be taken orally. For example, things like, oh my gosh, like hormone treatment therapy. So estrogen and progesterone commonly utilized in things like birth control, but also utilized in individuals going through menopause, right? So, or even post-menopause, if you have a hysterectomy or a hysterosalpingo-oophorectomy, which is where you have everything removed, hormones are administered and you can take those in an oral form. One of the main reasons we do that is since they're lipid soluble, they can get across the epithelial barrier of the intestines into the bloodstream. So they can do that effectively. Some hormones are not lipid soluble, can't dissolve through the cell membrane, and need to be injected directly into the blood supply. So things like insulin are a good example of that as a treatment for various kinds of diabetes mellitus. All right. So we've got another little infographic here representing humoral regulation, where, for example, up at the top, we've got low blood calcium levels. Then that triggers the activation of an endocrine cell when calcium levels are too low. Parathyroid hormone gets released from the parathyroid gland, which actually there's some more steps that go into that, which we'll discuss here in a minute. Parathyroid hormone gets released into the bloodstream. That will then migrate to wherever its target structure is. Turns out parathyroid hormone works really well on osteoclasts and increase osteoclast behavior. So then we increase the breakdown of bone so that we can remobilize stored calcium in that hydroxyapatite, reintroduce it back into the bloodstream, and then elevate our blood calcium levels, right? So then vice versa. If blood calcium levels are too high, then we can release or activate other hormones from other endocrine glands. We wouldn't release parathyroid hormone, we'd actually release the counter agent to parathyroid hormone, which is calcitonin, which would then inhibit the activity of osteoclasts and increase the activity of osteoblasts to reintroduce calcium into the bone, therefore decreasing calcium levels in the bloodstream. We can also excrete this through the kidneys if we need to, right? All right, so this is an example of humoral regulation, all based around chemical content within the bloodstream. All right. And then we've got neural control, which again, fairly self-explanatory. We all are pretty well versed in how neurons function. Action potentials travel down the axon, trigger an exocytotic event at the presynaptic terminal. Those release neurotransmitters or neuromodulators, depending on what they are. Then they can be released to wherever they need to go. In this situation, those neurotransmitters will then activate or create a excitatory postsynaptic potential, an EPSP, on the endocrine cell. So all that means is it's a go switch. It's like turning a light switch on once you get into a room. So then that endocrine cell gets activated with the introduction of these neurotransmitters, and then that endocrine cell or gland or whatever produces the hormones. Those hormones, again, since there's no ducts, they literally just exit the cell or the tissue via exocytosis. And since the capillary beds are pretty much saturating the entire gland, they can diffuse directly into the capillary beds. All right. So neural control. And then we've got these really weird mechanisms of hormonal control where we use hormones to regulate hormone production. Right. So there's some pathways that we'll talk a little bit about today, but we'll talk more about on Tuesday. And I encourage you guys to kind of read over the weekend, going over some of these different pathways, because some of them, involve multiple steps. And oftentimes the culprit is going to be the pituitary gland. So we mentioned the pituitary gland before. We talked a little bit about it last chapter. In chapter 8, when we talked about the nervous system. I mentioned it way back when we were talking about the skeletal system in chapter 6. So, now we're finally getting to the pituitary gland and what it does. This little gland, it has 4 different parts to it. It's got an anterior lobe, and the anterior lobe has 2 hemispheres, and then the posterior lobe has 2 hemispheres. So there's technically 4 pituitary nuclei in total. But what we really care about is just the anterior versus the posterior. And the difference is the anterior produces a lot of different hormones, and the pituitary doesn't really produce that much. It only produces 2, actually. And then we'll get into a little bit about glands that produce hormones versus glands that secrete hormones, which is there's 2 different ways to think about that. And then kind of where those hormones will then act on the body. So here in this figure, we've got these large green arrows that represent stimulatory pathways. So in hormonal control of hormones, there's 2 different kinds of hormones that we care about. So one of them is a releasing hormone. Called RH. different from Rh factor for blood types. And then we have inhibiting hormones, or IHs. So most of the hormones that are produced and secreted by the anterior pituitary gland actually require inhibiting and releasing hormones. Some hormones will act as inhibiting hormones on others, but there are dedicated releasing and dedicated inhibiting hormones too. So a common pathway that we're going to see is the hypothalamus has a bunch of different nuclei in it as well, but it's got little neurons that are connected to both the anterior and posterior pituitary. With the anterior pituitary gland, most of the hypothalamal neurons will release neurotransmitters that contain either releasing or inhibiting hormones. Those will then pass through a little stalk that we're seeing here called the infundibulum. I've got some other figures in here where that's written out, so you don't have to worry about like writing that down yet. But the infundibulum, it's also called the hypothalamal hypophyseal portal, potato. So the hypothalamus will release either releasing or inhibiting hormones, which will then have the corresponding effect on the anterior pituitary. If it's a releasing hormone, then the anterior pituitary is like, all right, cool. Then we can produce this particular hormone and then everything's cool. If it's an inhibiting hormone, then it's like, all right, then we need to stop. Okay, so that's pretty self-explanatory. So we then, let's say if this is a releasing hormone, that'll cause the anterior pituitary to release that particular hormone, which can do a couple different things. Sometimes these hormones will then just go out and act on target tissues, but sometimes these hormones will then go and act upon another endocrine gland, which will then produce its own hormones to then go and act on the target cell, right? So that's what this whole figure is showing here. So hormones produced by the anterior pituitary can sometimes go out and act on target endocrine glands, which then produce their own hormones that end up doing what they're supposed to do. All right, so let's talk about a couple different tropic interactions here where we have hormones that can either control themselves via negative feedback, or sometimes they can control themselves via positive feedback and enhance the effect of those particular hormones. So for example, let's see, so thyroid hormones inhibit the secretion of their releasing entropic hormones. So here, the anterior pituitary gland releases or secretes the tropic hormone, which travels in the blood to the target endocrine cell, okay? The hormone from the target endocrine cell travels to its target, but that same hormone that's produced will feed back and turn off the production of the tropic hormone here from the anterior pituitary, okay? So the hormone from the target endocrine cell also has a negative feedback effect on the anterior pituitary and hypothalamus, and therefore will decrease the secretion of the tropic hormone. So this tropic hormone kind of acting as the on and off switch right here. And then we have instances where we can have positive feedback. So this is what occurs during the menstrual cycle, during specifically the ovulation part of the menstrual cycle, where prolonged estrogen stimulus can increase the production of follicle stimulating hormone and luteinizing hormone during ovulation, all right? So the anterior pituitary will secrete a tropic hormone traveling to the blood and then to the target endocrine. Oops, that's not what I want to do. Not there yet. The hormone from the target endocrine cell will travel to its target. The hormone from the target endocrine cell also has the positive feedback effect on the anterior pituitary and increases the secretion of the tropic hormone. This can either be negative or positive feedback. Fairly self-explanatory. I know I've been talking a little bit fast, but how am I doing? Anybody have any questions, comments, concerns? Give me a thumbs up if I'm doing okay. If you guys want to ask any questions. All right, cool. Thanks, guys. Thanks, Mazen, Liz, Lucas, Kevin, Dane. I saw you give a thumbs up. All right. Okay. So next up, let's talk a little bit about how these hormones actually cause their reactions in the target tissue. So we're almost done for today, so we might actually end a little bit early, which could be kind of cool. All right, so let's say we've got our hormones floating around in the bloodstream. If we're lipid soluble, we're typically bound to some sort of a transport protein. If we're water soluble, sometimes we can be bound to a transport protein. Other times we can just kind of float around as needed. So every single hormone has a lock and key mechanism. They will only act on tissues that have the appropriate receptors or are able to allow for that hormone to diffuse through the membrane and affect the production of specific proteins within the cell. Okay, so there's always a lock and key mechanism that's present here. So even, like I said, even the ones that dissolve or can diffuse through the membrane have to go through transport proteins in order to do so. All right. So, like for example, you couldn't have insulin specifically. Actually, let me think about this. Insulin's a tricky one. Insulin has a bunch of random effects in the body. You couldn't have like glucagon, for example. If you introduce glucagon into, I don't know, I'm trying to think of something that doesn't have sugar in it. Let's say bone. Let's say mineralized bone. If you introduce glucagon into mineralized bone, nothing's going to happen, right? Because number one, it doesn't have the appropriate receptor on the surface of the bone cell membrane. But number two, even if it did, the bone cell itself isn't going to have any of the information inside of it that the glucagon hormone is carrying that's telling it what it needs to do. Hormones are essentially just these very intricate messengers that are constantly circulating through our bloodstream and can be produced at will via our various endocrine glands. All right. So now we're to kind of the last little bit of info we're going to talk about for today, which is our two main mechanisms in which hormones can act upon the body. So as I said, we've got either lipid-soluble hormones, which their basic pathway is they can diffuse through the membrane and enter the nucleus and then directly affect protein production at the source where DNA is at. Or we have hormones that can essentially create what I like to think of as like a giant Rube Goldberg, apparatus where you have, a domino effect that leads into a whole bunch of other really crazy, intricate things. If you guys want a really good example of a Rube Goldberg apparatus, look up the band OK Go on YouTube. They've got a bunch of really crazy Rube Goldberg apparatuses that they do for their music videos and stuff. It's really cool. But essentially, that's what water-soluble hormones do, is they create these large-scale cascade responses inside of the cell with just a single hormone that activates a particular membrane-bound receptor. So first we'll talk about lipid-soluble ones, and then we'll talk a little bit about our water-soluble ones. All right, so this figure pretty much summarizes it quite well. So lipid-soluble hormones, like I said, since they are fat-soluble, can dissolve through the cell membrane. Because remember, the cell membrane is a amphipathic structure. It has both polar and non-polar regions. The middle portion of the cell membrane is non-polar. So anything that is a polar molecule, like water or some of our water-soluble hormones, can't diffuse through that, right? So anything that is fat-soluble can get through there. So that's the key. Fat-soluble hormones, estrogen, testosterone, other kinds of androgens, progesterone, I think luteinizing hormone as well, that might be lipid-soluble. Anyway, these can dissolve through the cell membrane here. And their basic trajectory is that they will diffuse and enter the nucleus, all right? So now why the heck do they want to get to the nucleus? Well, the end result of every single one of these hormones in our body is to provide instructions for target tissues to produce proteins. That's the goal, right? So all of our hormones will initially strive to force target tissues to produce proteins as a response to, how do I want to say that? All of our hormones will initially strive to force target tissues to produce proteins as a response. Yeah. That kind of gets the point across. You can change the wording around a little bit, but all of the hormones that we're talking about, after they enter their target tissue, they do their thing. The ultimate response from just the hormone being produced there or acting on that tissue is for the cell to produce proteins in response. The production of proteins in response to that will look differently depending on whatever the structure is. So like, for example, in the liver, let's say that our blood sugar levels drop really, really low. The liver is one of the main sources that we can access stored sugar and fat in the form of glycogen. So glucagon will be released from the pancreas. It'll travel to the liver and then create a cascade response, because it's actually going to use the other mechanism that we'll discuss here in a little bit, to then create a whole sequence of breaking down glycogen and converting it into glucose, and then releasing that glucose into the bloodstream to elevate our blood sugar levels. So the initial response, though, is to turn on proteins, activate proteins, and then in some cases produce proteins, right? Whereas like here, things like testosterone or estrogen effects on target cells, or even things like growth hormone, will cause cells to either enlarge or produce proteins in response to this. And so we do that by directly affecting the two main processes for protein production, which are transcription and then ultimately translation. Lipid soluble hormones affect transcription. So really quickly, if you guys don't recall from way back in chapter 3 when we talked about transcription and translation, each of these different mechanisms for protein synthesis are important because they really determine what the ultimate structure of a protein is going to look like. So the first step is always transcription, where we convert our genetic information in the form of DNA to a usable blueprint of protein production called mRNA. You guys have probably heard of mRNA lately because it's been all over the news for the past few months, actually, with the coronavirus vaccine. And we'll talk a little bit about that more here in a second, too. So with our transcription effect, one of the ways I like to think about this is with our cells. Our cells are kind of like these little villages, right? So each of these villages, can work together with other villages. So cells being connected to other cells towards a common goal. And that goal for our cells is homeostasis, right? So within our villages, we've got this like kind of little big Capitol building on the top of this hill, right? And inside that building, there lives the super wise old monk who knows literally about everything, okay? So, but the problem with that is nobody out here who actually is responsible for the goings on inside the village can speak this guy's language or even knows what he's saying. So we've got a little series of guys that live inside the Capitol building here with our super wise monk that say, all right, cool. We can understand what you're saying. So what we're going to do is we're going to write down all the information that you've got, and not even necessarily all of it. We're just going to take the little chunks that we like from what you're saying and put it in usable information for the rest of the village. So these little writers or transcribers will sit down and write out useful bits of information that our wise old monk spits out, right? And the part of the problem is that this wise old monk just rambles and rambles and rambles and rambles. So even though we're parsing down everything that he's saying, this little bit of mRNA right here actually goes through further modification, especially in eukaryotes like us. Bacteria that do this, they don't actually have to do any editing because their wise old scribes are like, they're kind of dumb. They're not really dumb. They're just not as smart as ours are, really. So we've got to edit down and kind of splice out some of the information here. So we go through a little bit of mRNA editing before the final form of our mRNA molecule here gets sent out into the cytoplasm, ultimately for protein production. And that's where the rest of the village is like, okay, cool. We understand what this means. So now let's make an actual product from that. So then we've got our little ribosome protein factories that will then read out that mRNA sequence information and then start putting polypeptides together and folding up proteins, right? So where the hormones come into play is they are essentially, it's almost kind of like, or I don't want to switch up the analogies here, but they basically come in and tell the scribes like, all right, cool, you guys have been writing out all the stuff that he's been saying over here. We want you to write out this specific bit of information on this area of what he's saying. And they're like, all right, cool, we got it. So the hormone receptor complex here is basically an extra transcriptase enzyme that will then start to write out and transcribe a specific sequence of mRNA, right? So that is how lipid soluble hormones affect target cells, is they will affect and change up transcription processes that are occurring, and then ultimately change which proteins are getting made within that particular cell. All right. So with mRNA, really quickly, and you guys have probably heard about this, but there's one conspiracy theory that's been going around that, like, the coronavirus is gonna, or the coronavirus vaccine is gonna change our DNA, right? It's one of the common concerns that I've seen people voice, which, like, at first glance I can totally see why people would think that, right? Because they see mRNA, which kind of sounds like DNA, and we know that DNA is important because it's what makes us us, right? And if you're giving us new DNA, then aren't you changing our old DNA, right? Like, I can totally see how people would get there. But really, that's just completely not true with how our cells respond to mRNA, right? So what's nice about the vaccine and why it's so ahead of its time, and why it's so revolutionary in kind of how we develop vaccines, is because normally the way that our body works, and we'll get more into this once we get into the immune system, but essentially our body learns from prior infections because it'll get viral or bacterial DNA from being infected. And then over time, we will end up extracting that information, and we will make proteins as a way to protect ourselves from those other kinds of viruses and infections that could attack us again. But in order to do that, we have to get infected, and then we have to go through this whole process of fighting down or fighting back and phagocytizing that bacteria or virus or whatever, break it down, extract the information from it, from the the DNA, and then sometimes the RNA that the viruses will contain. And then we've got to make sense of that. We have to reverse transcriptase that and then reconvert that into a usable mRNA product so that we can make the proteins in order to do that. The cool thing about this vaccine technology is that we shortcut all of that. We have been able to reverse engineer the genetic information from some of the most common strains of the coronavirus and get just the mRNA sequence out of that particular viral capsule. What that does is then we can administer that into our bodies through a vaccine and then allow for our cells to pick up those mRNA transcripts and say, all right, cool. We can just make these proteins that we then stick on the surface of our cells that will then protect us from the virus, and in a way allow us to, if we become infected, fight off that virus before we even show any symptoms or before our body can succumb to the actual virus. So that's why this mRNA vaccine technology is so cool and so advanced, but also fundamentally why it's not changing our DNA, because DNA isn't even involved in that process. We've already got the stuff that we need to make proteins, so why involve the DNA at all? So in case you guys are wondering, or if you know of anybody who's been concerned about it changing our DNA, if you know anything about how transcription and translation work with protein synthesis, the DNA isn't even involved with mRNA, right? If we've already got the mRNA, or if we introduce ourselves to it, all right? So I know I got a little bit off topic there, but it's just too cool not to talk about. So with our lipid-soluble hormones, they control protein production in target cells, okay? So let's talk a little bit about water-soluble hormones. So these guys are a little bit different because they, again, sometimes they will result in the production of proteins, absolutely. But sometimes they can also just trigger intercellular communication mechanisms or responses inside of the cell, like inside liver cells, for example, to metabolize and break down stored fat and convert that into simple sugars like glucose. So sometimes it's a protein, but sometimes it's not a protein. But proteins are always involved. Okay. So this is one of my kind of favorite mechanisms that cells use to communicate with one another in a very quick fashion. And it's through something that we call a G protein signal cascade, because we utilize this G protein complex that has 3 subunits here called gamma, beta, and alpha. And it gets called G protein because in its inactive state, we have a guanine diphosphate molecule. So just like ATP and ADP, but instead of adenine, we have guanine thrown in there. That's what the G is representing. All right. So in normal inactive state, cells just kind of, plodding along, doing its thing. But then we've got this water soluble hormone that floats along and recognizes this particular receptor and fits in just like a lock and key. Okay. So then immediately what ends up happening is this receptor protein activates our G protein subunits over here, which then will convert GDP into GTP. So it gets replaced, all right, onto the alpha subunit of the G protein. So then what ends up happening is this G protein complex decouples from the receptor protein up here. And keep in mind too, this receptor protein can activate multiple G protein subunits at the same time. It's not just one. So what we'll see here in just a minute. So what ends up happening is this alpha subunit will dissociate from the rest of the G protein over here, because then it's going to migrate over to the other side of the cell membrane, which we'll see in a second, and it's going to activate an enzyme that's going to churn out a bunch of secondary signaling molecules that we call cyclical AMP. And that's important because it's essentially going to send out a really broad signal to the rest of the cell, and then we're going to activate a bunch of other proteins. I'm going to be super nerdy here for a second, but it's like in Lord of the Rings and the last movie in Return of the King, when Minas Tirith is under attack. If any of you guys watch Lord of the Rings, you'll know what I'm talking about. If you don't, you should watch it because it's a great series. But there's this whole sequence where all these like light towers and beacons get lit, and they travel across, Middle Earth to notify the rest of the humans that, like, holy crud, we're getting attacked by a bunch of orcs here in a little bit. And so essentially our G proteins operate the same way. They activate a signal cascade that starts out very small, but then creates a very large, widespread response as a result of just a single hormone activating a receptor protein, right? So as I mentioned, this alpha subunit here dissociates from the G protein complex, and then it's going to migrate over to another side of the membrane where we're going to activate our adenylate cyclase enzyme. So as I mentioned, adenylate cyclase does this thing where it converts ATP into CAMP, or CAMP, which stands for cyclic adenosine monophosphate. So we're just removing, instead of one phosphate from ATP, which would then be ADP, we're removing two phosphates, so it becomes AMP. But since the molecular shape is unstable in just a linear form, we actually create an extra covalent bond to create a cyclical phosphate group that sticks onto the adenosine molecule. So why is cyclic AMP used? Well, cyclic AMP is a very easy to produce and very volatile signaling molecule. Cyclical AMP will then convert or essentially activate other enzymes inside of our cells called protein kinases. Protein kinases are important because they trigger large, widespread intercellular responses that can activate things like glucose synthesis from glycogen, for example, if we're talking about glucagon. There's a whole host of other things that it can do depending on what you're talking about inside the cell, but we use cyclical AMP all the time. We can even use things like calcium content as well to activate, other kinds of intercellular responses, all right? So we can activate a bunch of protein kinases, which will then trigger an extracellular response. And then we can deactivate or convert extra cyclical AMP that's not being utilized back into AMP, which is an inactive form, via phosphodiesterase, which will deactivate cyclical AMP. All right. And then we can re-shove phosphate molecules onto our AMP molecule to transform it back into ATP or ADP if we need to. All right. So another way to think about this is kind of like I mentioned, this cascade response, large, widespread response from a single receptor hormone. We can activate several G protein complexes, which will then activate multiple adenylate cyclase enzymes, producing multiple cyclic AMP molecules. and then activating multiple protein kinases to create large, widespread intracellular responses. You can think of this as like the pay it forward scheme as well, where I shouldn't say it's a scheme because it's actually really, kind and good-hearted to, you help 3 people, each of those 3 people helps 3 people, et cetera, et cetera, et cetera. Or if you're a little bit more sinister, you can think of it in the reverse and think of it as like a pyramid scheme, right? Nice little Ponzi scheme going on right there. So that's the power of our water-soluble hormones, is that even though they can't diffuse into the cell to directly affect things like protein production via transcription, but they can create large, widespread cellular responses via this G protein signaling cascade. Okay, so let's see. We've got a couple minutes left here. So I'll just talk about some of the hormones that are produced in the pituitary gland, and then we'll be done for today. So pituitary gland, super duper important, produces a ton of different hormones. So as I mentioned, the posterior pituitary isn't really like as interesting. I'm going to put that in quotes because it is, but compared to the anterior pituitary, this thing only produces 2 hormones. So this is the difference here. The posterior pituitary gland will release these hormones because of the vascular network that's provided in here. But where the hormones are actually, I should say, let me back up. The posterior pituitary will secrete these hormones, ADH and oxytocin. However, these hormones are not produced in the posterior pituitary. They're actually produced via hypothalamic neurons up here, okay? So these axons that feed down into the posterior pituitary are the ones that will actually synthesize and produce the antidiuretic hormone and oxytocin hormone. However, the posterior pituitary gland is the one that secretes those particular hormones, if that makes sense. It's a semantics thing, but it's an important caveat to recognize that the posterior pituitary doesn't actually produce those hormones. It only secretes them. Whereas the anterior pituitary produces and secretes all of these different hormones right here. However, a lot of them will either be activated or deactivated via releasing or inhibiting hormones from the hypothalamus. So typically, if any one of these is going to get released, you have to have a releasing hormone coming from the hypothalamus to trigger the production of them. So we've got growth hormone, pretty much acts on every single component of the body, right? We have adrenocorticotropic hormone, which triggers the release of androgens and corticotropic hormones in the adrenal glands, in the adrenal cortex, right? We have thyroid stimulating hormone, which produces things like calcitonin in the thyroid. So you're quickly seeing here that these other endocrine glands that we see throughout the body respond to hormones that are produced by the pituitary. So we've got this kind of like roundabout network of hormones that are produced in the pituitary gland, but then go on and activate other glands within the body. We have follicle stimulating hormone and luteinizing hormone, which both are produced in the pituitary and both act upon the testes and the ovaries. So in the ovaries, this triggers the production of an ovum during the ovulation cycle. And then we have in the testes things like follicle stimulating and luteinizing hormone that are going to maintain the health of healthy sperm follicles and the health of the epididymis, but also the production of healthy sperm cells too. And then we have melanocyte stimulating hormone, triggering the production of melanin within the melanocytes of the epidermis. And then prolactin, which can trigger the development and maturation of mammary tissue within the breast. All right. But yeah, like I said, every single one of these is going to respond to releasing hormone and inhibiting hormone. So releasing and inhibiting hormone produced by the hypothalamus, and then produced and released by the hypothalamus, and then released into the anterior pituitary. But all of these hormones are produced and released by the anterior pituitary, not the hypothalamus. All right, I think that's good for today, right? What do you guys think?